Welcome back. In the first half of this chapter, we looked at what determines whether a chemical reaction is going to be spontaneous or non-spontaneous, what determines whether it's going to be an exergonic reaction as compared to an endergonic reaction. And we saw that endergonic reactions are actually converted into a series of exergonic reactions by coupling them with other exergonic reactions, such as the phosphorylation of a substrate by the hydrolysis of ATP. In this half of the chapter, we're going to think just about these spontaneous reactions and what determines how fast they're going to go. So even if a reaction is exergonic, if it's a spontaneous reaction, sometimes they can occur incredibly slowly. The example given in your book is that the hydrolysis of sucrose into glucose and fructose monomer units is an exergonic reaction. It gives up energy. But you can put sucrose into a beaker of pure water and let it sit there for hundreds, maybe even thousands of years without any appreciable change in the sucrose to those two monomer subunits. And so it's an exergonic reaction, but it's incredibly slow. And so in this web lecture, we're going to look at why are some of these energy releasing reactions so slow, what keeps them from just proceeding, and how in biological systems we increase the rate of those reactions. And we've been thinking about these biological reactions in terms of metabolic pathways. And we've got these little enzymes hovering over these arrows. So this time we're going to look at what are these enzymes, what are they doing there, what's their role in these metabolic pathways. So first, some definitions. A catalyst is a chemical agent that speeds up a reaction without being consumed by the reaction. So it takes part in the reaction, but it comes out of that reaction completely unchanged and ready to catalyze another reaction. So this is very common in chemistry. And in biology, we call these catalysts enzymes. So an enzyme is a protein that acts as a catalyst for chemical reactions. In the example I gave you before of sucrose being hydrolyzed down to glucose and fructose, the enzyme that facilitates that reaction is called sucrase. And so what are the barriers, what are the hurdles that need to be cleared that enzymes help with? So before a reaction can take place, reactants need to run into each other, they need to collide, and they need to collide in the precise orientation that's going to give them enough kinetic energy to break those covalent bonds that need to be broken in order to get the correct products. And once that happens, once those covalent bonds are broken, then we reach this very high energy, unstable, intermediate condition called the transition state. So remember that when atoms are covalently bonded, they have completed those valence shells. They are in a pretty stable state. Some covalent bonds are more stable than others, but in general, being covalently bonded is more stable than not being covalently bonded. And that intermediate, after the bond has been broken, before the new bond has been formed, is incredibly high energy and very unstable, and we call this the transition state. So the energy required to get over this hurdle, to be able to get up to this unstable high energy transition state, is called the activation energy. So the activation energy is required to strain the bonds of those reactant molecules so that they can reach this transition state, to break those bonds and reach this high energy state. So the reactions occur when the reactants have enough kinetic energy to reach that transition state. And so remember that the kinetic energy of molecules is a function of their temperature, so temperature is going to play a role in the rate of reaction. Thus, reaction rate depends on the kinetic energy of the reactants and the activation energy of a particular reaction, how unstable, how high energy is that particular transition state for that particular reaction. So the free energy change of the transition state. So looking at this in graphical form, we can see that we've got relatively stable reactants. So these are higher energy reactants than the products are going to be, but they are stably covalently bonded. They need an input of energy to break those bonds and reach this unstable transition state at relatively high free energy. And then once they've gotten here, then it's an easy downhill, spontaneous, completely exergonic process to get from here down to the product. But you need to get up this hill by the addition of energy. So how do enzymes help to solve this problem? So in catalysis, enzymes or other catalysts speed up the reactions by lowering the activation energy. So they're going to take this hump and make it a little bit smaller so that the kinetic energy that's available due to the temperature is going to be enough to make this reaction proceed. One thing to notice here is that the enzyme does not in any way affect the free energy change of the reaction itself. It just makes it easier to get over this hump to get from reactants to products. They don't change endergonic reactions into exergonic reactions. They just increase the rate at which reactions that would have occurred anyway, now they're going to occur faster. So enzymes are highly specific to particular substrates. So the substrates are the reactants that are going to participate in this reaction. And remember that enzymes are proteins. Proteins are extremely variable. These incredibly long chains of amino acids that can have highly variable primary structure that's going to give them highly variable shapes, that tertiary structure. So these are going to have very high specificity to their substrate. So when the enzyme binds to its substrate, in other words, the reactants, it's going to form a structure that we call an enzyme substrate complex. So this is the enzyme bound to its substrate molecule or molecules. So while they're bound, the activity of the enzyme is going to convert the substrate into the product. So a substrate goes in, a product comes out of this enzyme substrate complex. The reaction, not just the substrate, is very specific, but it's going to facilitate a very specific reaction. So the products will always be the same that come out of this enzyme substrate complex. And the place where the substrates bind, we call the active site of the enzyme. So you'll see a lot of images. I've posted some animations to the course website that shows this enormous enzyme with this tiny little substrate coming and binding to it. So it's going to bind at one very specific site that's going to have a high affinity for the substrate. Once that substrate binds to the enzyme, remember that a lot of times when proteins bind together or proteins bind to molecules, that can cause a shape change in that protein. And so in many cases with enzymes, when that substrate binds, the enzyme is going to change its shape a little bit to fit that substrate just a little bit better, to kind of snuggle it in and bring it right into the correct position to enhance that reaction. And we call this induced fit. The binding of the substrate induces the enzyme to change its shape to create that perfect fit. Here's an image showing that. You see the enzyme kind of spread out a little bit with this binding site wide open. The two substrate molecules come and find their binding site, and when they do, this enzyme is going to change shape and kind of close them in and put them just in the right positions to undergo that reaction. So what happens once they're snuggled into their particular enzyme? So the interactions between the enzyme and the substrate are going to lower the activation energy required for that reaction to proceed. And they're going to do this by stabilizing the transition state. So remember that enzymes are not consumed during the reaction. They're not changed in any way. They're not altered. They're going to come out of this in exactly the same form that they went into it. They just facilitate this reaction. And so they can be extremely fast acting, and they can come out of this reaction ready to catalyze another reaction. And so they can be used hundreds and hundreds and hundreds of times, thousands, maybe even millions of times. So a very small amount of enzyme can have a huge metabolic effect because they're used over and over and over again in these catalytic cycles. So here we have a visual representation of a model of how this can work. You can see on the enzyme there are binding sites that are very specific to the particular molecules that are going to fit there. In this case, we see just the right size and shape and distance from each other in the case of bound molecules. They're going to come and bind to their binding sites. So we have the first phase, initiation. Substrates are going to bind to the active site in a very specific orientation. The binding sites are going to put them in the right orientation relative to each other so that the correct parts of these molecules are going to be in contact that need to be reorganized. So this is going to form that enzyme-substrate complex. Number 2, transition state facilitation. So interactions between the enzyme and the substrate are going to lower that activation energy in some way. And then finally, termination. The products are going to be released after the reaction has occurred, and the products will have a lower affinity for those binding sites, and they're going to be released. So the enzyme is exactly the same at the end as it was when it started, ready to be used again. How does the enzyme lower that activation energy barrier? So first of all, just by having the substrates in the right positions relative to each other to be able to form those new bonds when the old bonds break. Another thing they can do is they can actually, with that induced fit, when the shape of the enzyme changes, it can change in such a way that it actually physically contorts that molecule and pulls on those bonds, straining them, making it easier for those bonds to break. It can create a favorable microenvironment. So for example, in the active site of the enzyme, perhaps in this particular reaction, a hydrogen ion needs to be removed from the substrate. And so maybe that active site includes a basic amino acid that's going to readily accept that hydrogen and pull that hydrogen atom away from the substrate molecule. Then, of course, it would have to give that hydrogen up again for the enzyme to not be changed, but it's going to give it up to the products. So that's one way that the enzyme itself can actually facilitate that reaction by providing a favorable microenvironment. And it can also temporarily, reversibly, covalently bond to the substrate to be able to have a different reaction with a lower activation energy to facilitate that transition. So what you're seeing here is a graph of substrate concentration. So as substrate concentration increases, how does that affect the rate of product formation? If you have an uncatalyzed reaction, remember it's likely going to be very, very much slower, but you're going to have a linear increase. The rate of reaction is going to depend only on how many reactants there are available to undergo that reaction. In the case of a catalyzed reaction, you're going to have a very steep increase in the rate of reaction up until a point where the enzymes are going to be completely filled with substrates. So when you get too high a concentration of substrates so that all of the active sites of the enzymes are occupied at any given time, then the reaction can't proceed any faster by adding more reactants. The only way to speed up that reaction at that point is to add more enzyme. And so we call this saturation of the enzyme. So all enzymes show this type of saturation kinetics, where if all of the enzymes are busy, adding more reactants is not going to increase the rate of reaction anymore. So active sites cannot accept substrates any faster. So no matter how large the concentration of the substrate gets, it's not going to go any faster. And those reaction rates are going to level off because all available enzyme molecules are being used. The only way to make the reaction go any faster at that point is to add more enzyme. The rate of enzymatic activity, the rate of the reaction when it's catalyzed, is also going to depend to some extent on the environmental conditions. So we know that protein shape is sensitive to temperature. We've talked about proteins being denatured, but also they can be folded into slightly different shapes at different temperatures. And so what we find is that enzymes tend to be optimized to work best in the conditions in their usual environment. So here we see different graphs for different bacteria that live in a cool and pH neutral environment as compared to bacteria that live in a hot and acidic environment. And we see the relative enzyme activity of a particular enzyme at different temperatures. And so we see that the rate of reaction for these bacteria that live in a cooler and pH neutral environment have their maximum rate of activity at a lower temperature as compared to the bacteria that live in a hot and acidic environment. So these enzymes have evolved to work their best in the environment where they typically occur. Same is true for conditions of different acidity, even within the same organism. So this could be enzymes that exist in your stomach as compared to enzymes that live in your small intestine are going to operate optimally at different levels of pH. So in this case, we're looking at the example of these bacteria from a hot and acidic environment as compared to bacteria from a cold and pH neutral environment. And we can see that these enzymes in the bacteria from an acidic environment have their fastest rate of activity at a much lower pH. We'd also see that in enzymes that work in your stomach, whereas enzymes from bacteria that live in a more pH neutral environment are going to have their optimal level of enzyme activity, their maximum rate of reaction at a much higher pH. And then in the case of things that live in a basic environment, such as your small intestine, this optimum is going to be even higher. So we can't have all of the possible chemical reactions going on in an organism at all times. It would not result in any kind of an organized, well-regulated biological system. We'd have chemical chaos if all of these enzymes were catalyzing all of these reactions all the time. So we need very precise regulation of these metabolic pathways to make sure that we get just the right amount of product in the right place at the right time, dynamically responding to changing conditions. So we've got systems of regulation that keep these reactions happening at the right rate, at the right time, in the right place. So a cell can do this by switching on or off genes that encode specific enzymes. So we can just synthesize the enzymes as we need a reaction to be catalyzed. Or these enzymes can be already produced and present in the cell, and then we can have different methods of regulating the activity of those enzymes. So there's a benefit to that because those enzymes can be there and ready to go much more quickly if we just have ways of increasing or decreasing their level of activity. So regulatory molecules might control when and where and how much an enzyme functions. So it may change the enzyme structure. It may change its ability to bind its substrate. And this can either increase or decrease the enzyme's function. We can have activators or inhibitors of an enzyme's function to get more when we need more product and slow it down when we don't need so much. So many enzymes are regulated by molecules that are not part of the enzyme itself, and sometimes they're not even organic materials. So things such as cofactors can be inorganic ions such as zinc, magnesium, and iron that are going to reversibly interact with the enzymes and change their activity. You can also have coenzymes that are organic molecules but are not part of the enzyme. They're going to interact with the enzymes, and we're going to see these two particular ones, NADH and FADH2, quite a bit as we move into cellular respiration and photosynthesis. There can also be other non-amino acid atoms or molecules that are permanently attached to those proteins that help to regulate its activity, and we call these prosthetic groups. So these regulatory molecules can be either inhibitors that slow down the activity of the enzyme, or they can be activators. If we think specifically about inhibitors, they can work in a couple of different ways. A competitive inhibitor is one that's going to occupy all or some part of the active site and is going to actually prevent that substrate from binding to its enzyme. So we can say that it's going to actually compete for space with the substrate. can also have what we call non-competitive inhibitors that are going to bind somewhere else on the enzyme and cause that enzyme to change its shape so that the active site is not available to the substrate. So not only can there be inhibitors that are part of the normal, healthy regulation of these enzymes, but there are also inhibitors that can be used as toxins, things such as sarin nerve gas that are going to either competitively or non-competitively reduce the activity of these enzymes. And so these are also commonly used as pesticides and antibiotics. So here's an example, a model of how competitive inhibition might work. Here's an enzyme behaving normally, bonding its substrates, and then here is one with a regulatory molecule that fits just fine into those active sites, but it does not participate in the normal reaction that would occur in that enzyme. So the substrates can't bind because that regulatory molecule is there preventing them from finding their active sites. In contrast to this competitive inhibition, we can also have the non-competitive inhibition, and we call this allosteric regulation. Allosteric means another site on the enzyme. So it can be used to either inhibit or stimulate an enzyme's activity. And an allosteric regulation occurs when a regulatory molecule binds to the enzyme at one site that's going to affect its shape at the actual active site. So it's going to affect the protein's function at another site. So most allosterically regulated enzymes are made from polypeptide subunits, so that quaternary structure that allows individual polypeptide subunits to work together as a functional protein. And each of those polypeptide subunits will have its own active site. So you can have more than one active site on a functional protein. And this enzyme complex is going to change its shape and oscillate between active and inactive forms. So there will be one shape that it can take where those active sites are available and are going to have a high affinity for their substrate, and then they're going to shift their shape spontaneously back and forth between that and another shape that's going to not have that active site available to the substrate. If we think of these shapes just kind of oscillating, going back and forth spontaneously between an active and an inactive configuration, an activator is going to stabilize the active form of that enzyme and make all of those active sites available to the substrate. So here we see an example of allosteric activation, where here we are in the inactive site, the binding sites don't quite fit the substrate molecules in this configuration. Then our allosteric activator comes and binds to this site, and that's going to stabilize the form in which the substrate fits nicely and has high affinity for the active sites on the enzyme. In the case of inhibition, so in this case, in the absence of the regulator, it's going to be oscillating back and forth between an active and an inactive form. Here we see the active form where the substrate molecules can go ahead and bind, but then if that regulatory molecule binds to that allosteric site, then it's going to stabilize this shape that has low affinity for the substrate and make those active sites unavailable to the substrate molecules. So here's a visual example of this. This is an enzyme that has a regulator that's a phosphate group, so it's going to be phosphorylated to put it into its active state. So this is the active site. We call this an activation loop that's going to bond with the substrate. And right now it's in sort of a loose, kind of extended position. But then when those phosphate groups come and bind to those active sites, then this active site is going to be put into a configuration where it's going to bind with those substrate molecules. Here's an example of those oscillating active and inactive states. So we can think of this as the active form where these active sites are open and ready to bind their substrate, and in this state they're kind of closed, those active sites aren't available, and so we can think of this activator in one of these multi-unit enzymes as kind of fitting in between, and so it keeps all of those active sites through the whole enzyme open and ready to bind their substrate. The inhibitor might fit in this configuration and stabilize it, keeping all of those active sites closed and unavailable. Cooperativity is a special case of allosteric regulation that can amplify enzyme activity. So this is going to be an allosteric activator. When the substrate binds to one of the active sites on one of the polypeptide subunits, that's going to stabilize the active form in all of the other subunits. So it not only makes one subunit active, but it keeps all of the other subunits in their open and active state, allowing an increase in the enzymatic activity. So we consider this allosteric even though it's binding to an actual active site, because the act of it binding to this active site is going to have an effect on the active sites elsewhere in the enzyme. So we still consider this allosteric regulation. Another form of regulation of enzyme activity is called feedback inhibition. And so in feedback inhibition, the end product of a metabolic pathway shuts down the pathway. It's going to inhibit some step earlier up in the pathway. When you've made enough of your product, you don't want to waste energy making more than you actually need, and so this is going to prevent waste of energy and resources, creating more product than you need. Feedback inhibition is an example of a really important concept in biology, a really important mechanism called negative feedback loops. And so this is an example of a negative feedback loop. This is a general diagram in which we have some sort of what we call a set point, an ideal value for whatever's being measured, and there's going to be some kind of a stimulus in the form of a deviation from that set point. Whatever the thing is, its level is not quite right. There's going to be some kind of a sensor that's going to monitor and detect that deviation, that change from the ideal conditions. And then there will be some sort of integrating center that's going to compare the current conditions to the set point. Then downstream from there, there's going to be some kind of effector that's going to cause a change to compensate for that deviation. So some kind of change that's going to bring it back to the desired levels. When that effector has worked, it's going to move the system back toward the set point, and we call this negative feedback. Why? Because an increase in some value is going to cause that value to decrease to bring it back toward some sort of set optimal condition. And so as you may recognize that this is basically what homeostasis is, right? Maintaining conditions within a stable range of optimal conditions for the workings of the cell. And this is primarily done through these negative feedback loops. So a more real-world, tangible example that you are probably familiar with is just climate control in a building. There's some sort of change in the temperature in the room. You're going to have a thermometer in the wall that's going to measure the temperature in the room and detect when it changes. Then there's a thermostat unit that's going to compare the temperature measured by the thermometer to whatever temperature you've set the thermostat to and see if it's too hot or too cold. So if it's below the set point, if it's too cold, the air conditioning is going to turn off, the furnace is going to turn on, so that low temperature is going to prevent the temperature from falling any further and create a response that's going to compensate for that low temperature. And so the response is that the room warms, the temperature increases toward that set point. The thermometer is going to check it again, compare it with the set temperature, and maybe it's gone too far, it's gotten too warm, now it's above that set point. At that point, the air conditioning is going to turn on, the furnace is going to turn off, the room is going to cool down and decrease toward that set point, and it's going to keep going through these different negative feedback loops to keep the temperature in the room exactly where you want it to be. So this is a very typical homeostatic mechanism to keep conditions within the cell right at their optimum. So in the case of enzyme activity, this feedback inhibition is going to occur when you get a high concentration of the product. So you've got your metabolic pathway here, going through different intermediates using different enzymes. And this end product is going to have an allosteric inhibitory site on the enzyme for this first reaction. So it's going to actually inhibit this first reaction in the pathway to prevent more product from being made. So when there's a very low concentration of the product, very little of it is going to find those allosteric binding sites, and the reaction will proceed. But as you start to get an accumulation of that product, more and more of them are going to interact with these inhibitory allosteric sites on this first enzyme, and it's going to slow down that reaction and inhibit more and more of these enzymes until there's just the right amount of product in the environment. So it's also really important for all of these enzymes, especially if they're part of a pathway, to be just in the right place to be able to encounter the correct substrates to catalyze their reaction. So these enzymes are going to be found in very particular locations where their particular reaction needs to occur. So structures within the cell help to bring order to these metabolic pathways to make sure they're in the right place at the right time to facilitate the right reaction. We briefly mentioned some cases where the enzymes are actually embedded into membranes to keep them in the right positions and working in the right order as you go through one of these metabolic pathways. But also in eukaryotic cells, these membrane-bound organelles are very important for keeping enzymes concentrated in the specific area where they need to be. So they're going to reside in very specific organelles. For example, enzymes for cellular respiration are located in mitochondria. And remember, we have these separate spaces between the inner membrane and the outer membrane, and also this matrix inside the inner membrane. These folds of the inner membrane are going to contain membrane proteins that are enzymes for metabolic pathways. So the matrix contains enzymes in solution that are dissolved in this cytosol that are involved in the second stage of cellular respiration. Don't worry too much about that at this point. That's coming soon. And then enzymes for the third stage of cellular respiration are going to be found embedded in these folds of membrane. So I want to wrap up this discussion by just thinking a little bit about how this multitude of complex enzymes, complex and very specific enzymes, came about in terms of evolution. So of course, enzymes are proteins. Proteins are coded by genes. And these genes, when there is a mistake in the replication of these genes, which happens on occasion, we call this a mutation, and that mistake in the copying of the gene can result in a change in the amino acid sequence in the protein. In other words, a change in the primary structure of the protein, which can cause a change in the activity of an enzyme. So these altered amino acids, particularly if they happen at the active site, can result in a new function. So this can now become an enzyme that catalyzes a new and different reaction. And if that new and different reaction is favorable in whatever environment the organism lives in, natural selection will maintain and increase the frequency of this mutated version of the gene. And you will have now, instead of one enzyme, two different enzymes that catalyze two different reactions. And so this happening over and over and over again in millions and millions and billions of organisms over millions and billions of years is going to result in this incredible diversity of enzymes that we see today. And so the example that you saw in your book, in the lab they were able to repeatedly mutate and select for a change in sugar metabolism in E. coli so that the beta-galactosidase enzyme can change the reaction that it catalyzes from metabolizing one sugar to metabolizing a different sugar. So if we think again about these metabolic pathways, you might wonder why? Why have this whole series of reactions going through all of these intermediates to get to a product? Why not just have a reaction that creates the product that you need? And it turns out that current thinking is that this might just be a remnant of the evolutionary process that led to this series of reactions. So the idea is that these metabolic pathways themselves evolved, and that the enzymes evolved to make these building blocks of life. The hypothesis currently is called retroevolution, and we call it retroevolution because it's a repetition of a backward process that's going to produce a multi-step metabolic pathway. So what that means is that the earliest enzymatic activity that evolved was the one that results in the final product at the end of our whole metabolic process. So there's some key molecule that's needed to be produced, and the first thing that would have evolved would have been the last step in the ultimate metabolic pathway that gives you that molecule that's needed. So originally in evolutionary history, there was a substrate that used an enzyme to create a product that was needed. Eventually, according to this hypothesis, the amount of this substrate, the concentration of this substrate would decline because it's all being used up to make this product. And so as the substrate got used up, if there was some sort of new enzyme, a newly evolved enzyme, that actually generates that more of that substrate, then we could get more of this product, right? And so the next thing that would happen would be the second to last step in our ultimate metabolic pathway that's going to generate more of this product so that it can go through this last reaction, become a substrate for this last reaction to get our ultimate product. So now we've got another substrate that's going to use the second enzyme that's going to create the substrate for the ultimate reaction that gives us the product that we want. But eventually this substrate is going to get all used up too, making all of these other products. And so then another enzyme evolves that uses a different substrate to create more of this substrate number 2 to be able to ultimately get to the final product. And so the cycle is going to repeat. Additional steps are added to the series of reactions until we end up with a metabolic pathway where there is a constant supply of the initial substrate from the environment that's going to be adequate to get as much of this product as we need. So we call this retro because it begins with sort of the end. The evolution begins with the end of the pathway and adds the next previous step in a backwards direction until we get this long, complicated, multi-step pathway. In this web lecture, we've seen how enzymes help to facilitate and speed up reactions that would have occurred anyway, exergonic reactions that are spontaneous but very slow, and how important this is to life's processes. We've seen how these enzymes are regulated to make sure that we get the right amount of product at the right time in the right place. And so now we're going to use all of these tools in the next segment to be able to understand the metabolic pathways using enzymes that lead to the production of ATP, which is the ultimate currency that cells use to be able to do work.